This QuantiGene training tutorial is designed to illustrate 3 key steps, which include overnight incubation of the samples with the magnetic beads using the VorTemp instrument, washing of the magnetic beads in the magnetic separation plate using the handheld magnet, and incubation of the preamplifier with the magnetic beads. The sample type for this tutorial is tissue homogenates. Prepare an appropriate volume of the working bead solution containing nuclease-free water, lysis mixture, blocking reagent, proteinase K. Prior to adding the magnetic capture beads, vortex the vial for 30 seconds. Add the 2.0 probe set and vortex for 15 seconds. Transfer the working bead solution to a plastic reservoir and pipette 60 microliters of reagent into each well of the hybridization plate well that will be assayed. Add 40 microliters of homogenizing solution to 3 wells to act as background controls. Add 40 microliters of samples to each well. Seal the plate using the provided pressure seal and apply firm, even pressure across the plate using a soft rubber roller. Prior to incubating the hybridization plate, place an inverted plate lid into the VorTemp and calibrate the temperature for 54 and 50 degrees using a temperature validation kit from Affymetrix. Place the hybridization plate onto the inverted plate lid and incubate overnight for 18 to 22 hours. Set VorTemp to 54 degrees C and 600 RPM. After incubation, spin the plate in a centrifuge to collect all of the reagents to the bottom of the wells. Carefully remove the pressure seal and pipette the contents up and down 5 times, then transfer to the magnetic separation plate. Once transferred, place the magnetic separation plate into the handheld magnet and lock the plate into place with securing tabs. Ensure that the plate is locked into place by pulling on the 96 well plate. Wait 1 minute to allow the magnetic beads to accumulate on the bottom of each well. To wash the beads in the plate, quickly invert the assembly over a sink and gently blot onto several layers of paper towels to remove any residual solution. Pipette 100 microliters of 1X wash buffer into each well using a multi-channel pipette and wait 15 seconds to allow the beads to accumulate on the bottom of the well. Quickly invert the assembly over a sink and gently blot onto several layers of paper towels to remove any residual solution. Wash the plate a second time by pipetting 100 microliters of 1X wash buffer into each well and wait 15 seconds to allow the beads to accumulate on the bottom of the well. Quickly invert the assembly over a sink and gently blot onto several layers of paper towels to remove any residual solution. Wash the plate a third and final time by pipetting 100 microliters of 1X wash buffer into each well and wait 15 seconds to allow the beads to accumulate on the bottom of the well. Quickly invert the assembly over a sink and gently blot onto several layers of paper towels. Proceed to the next step of adding 100 microliters of preamplifier working solution to each well. Seal the plate using the provided plate seal and apply firm, even pressure across the plate using a soft rubber roller. Place the plate onto a Microtiter plate shaker and shake for 1 minute at 800 RPM to ensure that the beads are resuspended. Ensure that an inverted plate lid is seated in the VorTemp, and then place a magnetic separation plate into the VorTemp. Incubate for 1 hour at 600 RPM at 50 degrees C. Preparation and incubation of the amplifier, label probe, and SAPE are similar to the steps just described. This concludes today's video tutorial for use with the QuantiGene Plex assay.